Hello friends, Ace Botane next episode. In this episode, we'll be discussing about molecular basis of inheritance, 11th one, that is the last one. Mainly fingerprinting we'll be discussing. So we can proceed. So for convenience, I will share the screen. I think screen came. So we can proceed. Molecular basis of inheritance, 11th one, last one. Which all topics we'll be discussing in this episode. One, as usual, introduction. Then principle and some basic terms in connection with the fingerprinting. Then some basic steps in fingerprinting. Then as usual, 5 theory question, 5 NEET model question with the key. So we can proceed with the introduction. So most basically, when we say fingerprinting, what it means? It means a technique connected with the DNA by which we can identify each individual among humans. Because fingerprinting or fingerprint is unique to each individual. That is the most simplest manner we can explain what is fingerprinting or what is the purpose. Now, initial work of it is connected with the famous scientist Alex Jeffrey in 1984. Photograph is given there. And he discovered the technique of genetic fingerprinting. In India, the most famous institute connected with the fingerprinting technology of fingerprinting is CCMB. That is the Center for Cellular and Molecular Biology, Hyderabad. And father of Indian DNA fingerprinting is the famous Laljit Singh. He was passed away in 2017. So birth and that year where he born and dead, that is given there. Now we can proceed to next page. I think first page is clear. Now comes second page. So DNA fingerprinting, it is like a synonym. We can use two terms, almost same as DNA profiling or even DNA typing. Even though slight variations may be there, that is why I use the term almost same as DNA profiling or DNA typing. Now, what is the principle of DNA fingerprinting? See, DNA fingerprinting is a method by which each individual can be distinguished from other or others. And we already know 99.9% of the base sequences among humans are same, clearly given in CRT. So in a mathematical way, when we put it, grand total is 100%. Total base sequence similar in humans is 99.9%. That means variation dissimilar will be 0.1%. So what will be this 0.1%? That is equal to 0.1% is equal to 1 by 1000. So it will be 3.3 into 10 raised to 9 divided by 1000. So it will be equivalent to 3.3 into 10 raised to 6 base sequences will be different. Simple mathematical logic. Interestingly, these base sequences, differences can occur in genes and repetitive DNA. Identifying this different base sequence in some specific regions in DNA sequences known as repetitive DNA, and this is the basic principle of fingerprinting. So it is connected with the repetitive DNA. So identifying these different base sequences in specific regions in DNA sequences that will be repeated. So it is called repetitive DNA, and this is the basic principle of fingerprinting connected with the repetitive DNA. I think idea clear. Now we have to proceed with some basic terms, because why I'm putting in this manner, so students will be feeling comfortable and easy step by step. Bulk DNA, repetitive DNA, satellite DNA. Bulk DNA means the term indicates majority of genomic DNA containing a mix of unique sequences that are not significantly repeated. So bulk DNA means majority of genomic DNA containing a mix of unique sequences that are not significantly repeated. While repetitive DNA means specific DNA sequences that are repeated multiple times within the genome. It will often appear as distinct bands separate from the bulk DNA. So repetitive and bulk DNA. I think it is clear. Now we can proceed to next page. So it will be often appear as distinct bands separate from the bulk DNA. Next page we can proceed. When separated by density gradient separation. As continuation, bulk DNA is the functional non-repetitive sequence, and repetitive DNA is the sequence that repeat frequently within genome, forms a large portion of human genome. So more functional is bulk DNA, which is non-repetitive sequence. While repetitive DNA is a sequence that repeat frequently within the genome, form a large portion of human genome. That line is given in NCERT page number 105. Bulk DNA forms major peak, and the other small peaks are referred to as satellite DNA in density gradient centrifugation. And remember, satellite DNA sequences normally do not code for any proteins. Again, NCERT page number 105, because this type of statement they're asking. Repetitive DNA and satellite DNA are practically the same. Satellite DNA is a type of repetitive DNA. I think that three items clear. That is bulk DNA, repetitive DNA, satellite DNA. Now again, we have to proceed with certain terms. VNTR, mini satellite, SSR, microsatellite, DNA polymorphism, and last term is PRO. Because certain basic terms in fingerprinting, then only we can proceed to the method and steps. So classification of satellite DNA, basic criteria. Repeating, that DNA is called satellite DNA. What is the basic criteria for classification of satellite DNA? Basically, three items connected with NCERT. One, base composition, whether A to T rich or G to C rich. Second is length of the segment, and third is number of repetitive units. Now we have to proceed with the VNTR. Full form is variable number tandem repeat. This is also called as mini satellite and found in genome of many higher eukaryotes. Mini satellites surrounded by conserved restriction sites. It is a small DNA sequence tandemly arranged in many copy numbers. Repeating, number is variable in an individual from chromosome to chromosome, and VNTR will show high degree of polymorphism. What is polymorphism? We'll be studying later as we proceed. And size of mini satellite or VNTR varies from typically, it's a popular question, 0.1 to 20 kilobase pairs, KB. That is regarding VNTR or mini satellite. Now SSR, single sequence repeat. Other name is STR, short tandem repeats. Other name is microsatellite. And number of base pairs will be usually very less, 1 to 6. I think mini satellite and microsatellite is clear. Number of base pairs, they're synonyms, important for exam. Theory, NEET. Now comes the term DNA polymorphism. So when we discuss the term polymorphism, prefix is poly. Poly, many area we know, even polymer. So poly is connected with the Greek language. Poly means many, more, multiple, etc. Cell cycle, there is a cell organ, lysosome is a polymorphic cell organ. Animal side, obelia is a polymorphic colony. Same manner, what is meant by DNA polymorphism? Because it's the basis of fingerprinting. Most basically, when we analyze DNA polymorphism, it is the natural variation in DNA sequences that exist between individuals within population. So variation in DNA sequences between individuals within a population. For this, DNA from tissue, blood. So normally every tissue, blood, hair, follicle, skin, bone, saliva, sperm, etc. shows same degree of polymorphism. And all this same degree of polymorphism within an individual. I think polymorphism is clear. So natural variation DNA sequences that exist between individuals within a population. And DNA from every tissue in an individual like blood, hair, follicle, skin, bone, saliva, sperm, etc. shows same degree of polymorphism. So it is specific to an individual. Further, different people have slightly different DNA sequences at certain locations. It is the occurrence of multiple different forms of a specific DNA sequence within a population. So variation. Most common type is single nucleotide polymorphism, which is otherwise called SNPS. In humans, scientists have identified 1.4 million locations where SNPs, single base DNA difference, occurs in humans. That statement given in NCERT page number 104 in last episode, we passed through it in Human Genome Project. I think page is clear. Now we can proceed to next page. Now, polymorphism is the basis of fingerprinting. So why DNA polymorphism is so important? DNA polymorphism is the basis of genetic mapping of human genome as well as DNA fingerprinting. Again, the statement in NCERT page number 105. Now, once again, we are proceeding to DNA polymorphism because it is an important even basic term in fingerprinting. Polymorphism arises due to mutations. Mutation, we already know, sudden drastic irreversible changes. Generally, when we speak mutations, variation in genetic level. New mutations may arise in an individual, either in somatic cell or in germ cells. Germ cells means cells connected with the gamete production. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutations, this way it can spread to the other members of the population through sexual reproduction. Allelic sequence variations, as normally called as a DNA polymorphism. Again, slight variations in allelic sequences. Now, DNA polymorphism, if more than one variant, that is allele, at a locus, occurs in human population with a frequency greater than 0.01. 0.01 means 1 by 100. So variations, that is the basic meaning. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. That's the basic, most simplest manner. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. Probability of such variation is observed in non-coding DNA sequences would be higher as mutation. Once again, the probability of such variations to be observed in non-coding DNA sequences would be higher as mutations. Next we will take. As mutations in the sequence may not have any immediate effect or impact in an individual's reproductive ability. Now, what is the effect of all this? These mutations keep on accumulating generation after generation and form one of the bases of variability or polymorphism. There are different types of polymorphism ranging from single nucleotide change to very large scale changes. And for evolution and speciation, such polymorphism play a very important role. That is how the variation comes, that it becomes the base of fingerprinting. Now comes the term prop. Prop, in our case, or that generally means a short single-stranded DNA sequence that is labeled with a radioactive or a chemical tag used to identify specific complementary DNA sequences within a sample by binding to them essentially as a marker to detect a particular genetic region in DNA fingerprinting. So we are discussing DNA fingerprinting. That is how probe can be defined. RNA can also act as a probe, single-stranded, then depending on experiments. So RNA can also act as probe. It will be also single-stranded. Depending on experiments, DNA or RNA can be used as probe. Now I put some testing events. NCERT not dealing it except one. Anyway, we can proceed. One is Southern blotting. Southern blotting for identifying specific DNA sequences in a complex mixture. DNA fingerprinting, it is a base. Western blotting, specific proteins in a sample that is concerned with immunogenetics, etc. Northern blotting, RNA sample detection. Then connected with again gene expression, etc. Eastern blotting to detect post-translational modification of proteins. But it is variation from Western blotting. So 4 types of blotting just mentioned for us only. Southern blotting is needed. Now, basic steps in fingerprinting. Here I put some diagrams from NCERT itself. Some previous editions also I put. This is the present edition NCERT diagram. Now, I enlarged it a little bit here. And another one. This will not be available in your present edition of NCERT. This is previous edition of NCERT diagram with the steps shown. Everything mentioned step by step, they mentioned it. Now, next page we can find few more diagrams. These all are previous NCERT diagram. This is human genome. Present NCERT not given. It's the final result of fingerprinting. And this is what is known as VNTR, mother's, father's chromosome, etc. And how they are able to identify it likewise. Now, what are the basic steps? Steps already given here, but we can proceed in another manner. So what are the basic steps in DNA fingerprinting, how it will be proceeded? Everything with a simple manner, I put it there. See, first step is extraction. Extraction and isolation of DNA from a source. Diagram also you can watch here, diagrams here. Then cut into fragments by restriction endonucleases. Then DNA fragments by gel electrophoresis, which is otherwise called RFLP, restriction fragment length polymorphism. That itself is a question for expansion, are separated according to the size. So extraction, isolation, cut into fragments, restriction to nucleases, gel electrophoresis, RFLP, then it is separated according to the required length. These fragments will contain VNTRs. There is a technique you may be studying in zoology, PCR, polymerase chain reaction technique, also used to get more copies. Then after this gel electrophoresis, they are treated with the alkaline substances to get single-stranded DNA. Single-stranded DNA will be getting by treating with the alkaline substances. Now, separated single-stranded DNA fragments transferred into a nylon or nitrocellulose membrane, fixed on it by giving suitable required condition like temperature and time, etc. And this procedure is called Southern blotting by E.M. Southern, 1975. Now, the nitrocellulose with single-stranded DNA made into contact with the radioactive DNA probes, having repeated base sequence complementary to possible VNTRs. The nylon membrane is washed to remove extra probes. An X-ray film is utilized to mark the places where the radioactive DNA probes have found to be on DNA fragments. These places are marked as dark bands when X-ray film is developed, that is called autoradiography. And the last step is one. The dark bands on X-ray film represents the so-called DNA fingerprints. Diagrammatic way, this is the representation as original, that is a radiograph. I think it is clear step by step, compare with the diagram and proceed. Now comes application of DNA fingerprinting. This is originally taken from a previous edition of NCERT, application of DNA fingerprinting. The technique is now used to identify criminals in forensic laboratories, determine paternity, then who is the true biological father or mother, verify whether hopeful immigrant is or as he or she claims, really a close relative of already an established resident, and identify the racial groups to rewrite biological evolution. So many applications are there. After that, as usual, 5 theory question, 5 NEET model question with the key. So what all things we studied in this episode? See, in this episode, we studied only one item, last item of this molecular basis of inheritance, namely fingerprinting. Since it is a little bit big, that is why I put it as a single episode. Now, introduction, identifying each individual, Alex Jeffrey, as well as the famous Indian fingerprinting scientist Laljit Singh. Then, synonym of DNA fingerprinting, profiling or DNA typing, almost same. Then, principle of DNA fingerprinting, 99.9% same in all humans, base sequence, only 0.1, and identifying this 0.1. Now, some terms like bulk DNA, repetitive DNA, satellite DNA, majority of genomic DNA, mix of unique sequences, not significantly repeated. Repetitive DNA means multiple times repetition will be there within genome. Then it appears as a distinct band separate from bulk DNA. Now coming to next one. And separation is made by density gradient separation. Now bulk DNA functional non-repetitive sequence. Bulk DNA forms major peaks. Then repetitive DNA is the sequence that repeat frequently within genome, forms a large portion of human genome, and that is otherwise called small peaks or refers to be satellite DNA in density gradient separation or centrifugation for satellite DNA. And satellite DNA sequence normally do not code for any protein. Repeated DNA and satellite DNA are almost same. Then VNTR, mini satellite, SSR, microsatellite DNA polymorphism prop, classification based on base composition, length of segment, number of repetitive units. Then after that, we entered into the term DNA polymorphism, most basic of fingerprinting. Polymorphism means variation. So polymorphism is natural variation DNA sequence that exists between individuals within a population. And that variation in DNA sequence within an individual will be same. Then most common type is single nucleotide polymorphism, SNP. And why DNA polymorphism is so important? Because it is the basis of DNA fingerprinting. Then simple terms how we can define DNA polymorphism. Most basically, DNA polymorphism means if more than one variant allele at a locus occurs in human population with a frequency greater than 0.011 by 100. Or more simple term, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. Probability of such variations to be observed in non-coding DNA sequence, that would be higher. Because mutations in the sequences may not have any immediate effect or impact. Next is prop. Prop means a short, single stranded DNA sequence that is labeled with a radioactive or a chemical tag used to identify specific complementary DNA sequence within a sample by binding to them, and is the basis of fingerprinting. RNA can also act as a prop, single stranded, but depending on the experiments, DNA RNA can be used as prop. Now, 4 types of blotting. Then our diagram starting, NCRT diagram. It is a little bit enlarged. And here I can find this sample tallies. Here I can show you. This sample, same pattern, tallies with this sample. This is how fingerprinting is done. Clearly, we can identify. Then different steps. That is given here. Previous edition printed there. Then human genome. Then different aspect. After that, steps I given there. Different steps, step by step. And finally, applications of DNA fingerprinting with a 5 theory question, 5 NEET model question with the key. If you like this episode, subscribe all, like it, share it, comment on it. We can meet in the next one, next episode with the second year, a new chapter. So by this, we finish second year, sexual reproduction, flowering plants, principles of genetics, molecular base of inheritance. So 3 big chapters. The remaining chapters are more theoretical and easy. So hoping that it is clear, we can meet later. Take care. Bye. Thank you.